Check 1, 2. You can just flip that on, and then
Good morning, everyone. I am Brenda Weigel. I'm not David Largaespada. But it is my immense pleasure to welcome you to the 28th, I can't believe it, 28th annual Nesbitt Lectureship in honor of Dr. Mark Nesbitt, who joins us here today. I'm going to take just a couple minutes to tell everyone a little bit about Mark and about some of the other very special people who are part of the history and legacy of the Pediatric Hematology, Oncology, and Blood and Marrow Transplant program here at the University of Minnesota. And when I talk about Mark, I would not be talking about Mark without mentioning his wonderful wife, Deb. They are a dynamic duo. And the reason Mark was so successful, and everyone will tell you this, is because they did it as a team. And so when I'm honoring Mark, I'm also honoring Deb, who will be joining us tonight at a wonderful dinner in Mark's honor. Mark was truly a pioneer in pediatric oncology. Mark joined the University of Minnesota faculty in 1959 and rose very quickly to the ranks of full professor within 6 years. He became section head in 1979, a position he held for 24 years. He also became the associate clinical director of the Masonic Cancer Center, which I will show you a picture later, was led by another pediatric oncologist, Dr. John Kersey, as the first head of our Masonic Cancer Center. Mark was pivotal in building and growing what we now take for granted in pediatric oncology, which is a culture of clinical trials and clinical research. Mark led the first trials in pediatric leukemia improving outcomes, establishing that we need to use intrathecal or spinal administered chemotherapy to cure children with leukemia. Mark also led the first national trial for Ewing sarcoma. And this really paved the way for the use of chemotherapy in combination with surgery and radiation for children with Ewing's. He published more than 300 papers in his career and trained more than 100 pediatric hematology, oncology, and blood and marrow transplant fellows and mentored many, countless of us in our careers. And I think this is probably one of the things Mark is most proud of, are the trainees that he was privileged to work with over the years. Mark was the true triple threat and leader in the field. He not only led here at the University of Minnesota, he led nationally and internationally. He was very active in establishing the first treatments for histiocytosis and was on the board and actually received an Outstanding Investigator Award from that society for his tremendous work and leadership in the field. He really was the founding member of the Children's Cancer Group, which is now the Children's Oncology Group. Without Mark, the systems that we now have in place would not exist. And this is a real incredible legacy to the field of pediatric cancer. He was a president of the American Society of Pediatric Hematology Oncology and the very first U.S. president of the International Society for Pediatric Hematology Oncology. I did not say that in French, but I should have because it is actually in French. And he was honored to receive an Outstanding Achievement Award from the American Cancer Society. Not only was Mark a leader in research and academia, he was a leader in the community and establishing advocacy and establishing several of the institutions we now take for granted. He was on the board of Make-A-Wish. He, was past vice president of Combined Health Appeal for Minnesota, and very importantly, in partnership with Dr. Norma Ramsey, really founded the Ronald McDonald House here in the state of Minnesota. This was really a huge undertaking for them at the time and a legacy to be proud of. And he is one of 6 physicians honored by community caregivers, by Minnesota physicians, a real pillar not only in the community of medical oncology and pediatric oncology, but a pillar in the community of advocacy for children with cancer. Mark really led a crew at the time that was amazing and really leaders in the field. In this picture, you will see Dr. Norma Ramsey up there on your right, who's sitting right over there, Mark in the middle. Diane Arthur on the right was really one of the, on your left, was really one of the pioneers in genetics for pediatric cancers. Across the bottom is Dr. John Parenthesis, who's now head of pediatric oncology at Cincinnati Children's. In the middle is Dr. Bruce Blazar, someone you know and love. And Bill Woods is on the right bottom corner, who is still practicing at Aflac Children's Hospital in Atlanta and was head of the program here for quite a while. And this is the, I would say, incredible trio. Norma on the left, who headed the bone marrow transplant program, Mark in the middle, who headed the hematology oncology program, and John Kersey on the right, who was the first head of the Cancer Center and a pediatric oncology, a pediatric oncologist. We could not have had a greater trio to lead not only pediatric cancer, but cancer here at the University of Minnesota. But Mark did this for the kids. Everything Mark did was centered on the children and improving the outcomes for children with cancer, which he really did, not only by establishing therapies, but by establishing the system, which is now the Children's Oncology Group, that has truly been transformative in the care of children. And we all are building and carrying forward that strength that Mark established, not only a strength of caring for children and focusing everything on them, but really caring about building the future through trainees, through his work, and through building a system that we now use to move forward. So it is my tremendous honor to introduce Dr. David Largaespada, who will give this year's 28th Nesbitt Lectureship. And I'm going to really mess this up. Probably because, okay, I've got to move to the other slides. So David, here we go. David, I'm not going to mess you up.
You can hit that play button.
Can I hit hang on. Hit the play. There we go. Play slideshow. There we go.
That'll work.
So David is a PhD scientist in our division of pediatric hematology oncology. He's a professor not only in pediatrics, but in molecular genetics, and has really focused his career in understanding the genetics of cancers, and now most importantly leads our pediatric brain tumor program, and has really established one of the world's leading programs in understanding cancer predisposition processes, particularly neurofibromatosis type 1. So it is my tremendous pleasure to introduce Dr. David Largaespada.
Well, thanks so much, Brenda, and thank you, Dr. Nesbitt, for being here. It's an honor to get to give this lecture. Well, thank you. So at the beginning, I just have to show you some disclosures. The university requires that I do this, so I've started some biotechnology companies, and some of our work is funded by Genentech, and work also with a local biotechnology company called Surrogen that makes porcine models of human disease, but I won't be discussing work related to these companies today. What I do want to talk about instead is how identifying cancer driver mutations has had a profound impact for therapy for cancer. And one of the classic examples that you all probably know about is in chronic myelogenous leukemia, where the discovery that almost all CML cases were characterized by the generation of a fusion oncoprotein called BCR-ABL led to the identification of a drug called Gleevec, which inhibits the activated kinase, Abelson kinase, and is tremendously useful for treating this disease. And there are other examples that we can point to as well. For example, in breast cancer, the identification of the HER2 oncogene, or oncoprotein, as a driver of breast cancer development and aggressiveness led to the development of a humanized monoclonal antibody called Herceptin, which can bind to and inactivate HER2, and this is useful for treating breast cancer cases that are characterized by HER2 amplification. So this kind of concept has led to the idea of precision medicine, that we could look at the genome of a cancer cell and try to decide what therapy we should use based on the alterations that are driving that cancer. And in fact, cancer gene identification is an FDA-approved concept for precision medicine in cancer. For example, this company, Foundation Medicine, has a test called the Foundation 1 CDX test, an FDA-approved genomic profiling test that you can use for all solid tumors to try to help decide the diagnosis, the appropriate diagnosis of a case, and what therapy might be useful. So this all sounds good. However, this idea of precision medicine has not benefited some patients at all. So for example, some childhood tumors, some sarcomas, they're not really, we can't really benefit from this precision medicine idea. And an example of this is highlighted by this patient, Zachary Bartz. He had neurofibromatosis type 1 syndrome. He and his family were champions for NF1 research in Minnesota and great friends of ours. He suffered from optic pathway glioma, but tragically he passed away at age 11 from an aggressive sarcoma. And his disease really exemplifies for me some of the challenges that remain in treating childhood solid tumors. We can't easily treat disseminated disease. These sarcomas don't typically respond to the immunotherapies we have. But importantly, a genomic analysis of these types of sarcomas has revealed few clues about how to treat them. And in fact, if you look at pediatric solid tumors in general, very few of them have so-called druggable targets. So this paper was a publication on the Individualized Cancer Therapy, or ICAT study. And they evaluated 100 pediatric solid tumors by whole exome sequencing and copy number analyses. And there was matched targeted therapy in only 3 cases. Those 3 cases got their matched targeted therapy, but there were no objective responses. So basically what I'm saying is that with today's knowledge, there's few actionable mutations in many pediatric solid tumors. Another more recent paper from last year in Nature did a fairly extensive genomic profiling of many forms of pediatric cancer. Some of them have fairly high frequency of so-called druggable target alterations that you can see here, for example, in MAP kinase signaling or PI3 kinase signaling, but few of these have actually been validated as really druggable. So that's why they said they're potentially druggable. So what do we do about treating cancer that doesn't have these actionable genomic mutations? This is something I'm interested in the lab work that we're doing here at the university. And so I'm going to tell you about 2 ideas, 2 approaches that we're taking to try to address this problem. And they're based on 2 different ideas. One is that there are useful drug targets in these tumors, and they can be found in copy number or epigenetically altered oncogenes. So we just have to figure out what these genes are. And I'll tell you an example from osteosarcoma that we've pursued. And the second idea is that while these tumors are not characterized by obvious activated oncogenes that we can drug, they are characterized by the recurrent loss of certain tumor suppressor genes. That happens over and over again. And it's possible that the loss of these tumor suppressor genes uncovers vulnerabilities that we can exploit for therapy. So this idea is called synthetic lethality. So the effects of tumor suppressor gene loss may provide new avenues for treatment. And this concept is an old concept that goes back to Drosophila genetics and yeast genetics, where it was found that the loss of certain genes is viable for the cell or the organism, but certain genes that have a synthetic lethal relationship, when lost together, result in cell death or death of the organism. if one of these genes is a tumor suppressor gene, we can find the genes that have a synthetically lethal relationship with the tumor suppressor gene and exploit that for therapy. All right. And I'm going to give you an example of this line of research that we're pursuing in malignant peripheral nerve sheath tumor development and treatment. So what about this first idea that there's useful drug targets found in copy number or epigenetically altered oncogenes? So an example of this is osteosarcoma. They seem to have few recurrent mutations and no obvious targets. This is an older paper from 2014, but the basic conclusions are still true. They resequenced whole exome and genomes and the RNA of human osteosarcomas, and the only gene that was reproducibly mutated at high frequency was P53, although there were some compelling mutations that occurred at low frequency, such as loss of P10 or activation of PI3 kinase. In fact, some of the osteosarcomas had no obvious driver mutations at all. And very few activated oncogenes were found. So it's not obvious from this how we find useful drugs for osteosarcoma. And this situation we call the missing drivers problem. So what is it that really causes these tumors when there are no obvious oncogenes? Are they caused simply by the loss of a few tumor suppressor genes? What's the nature of the epigenetic or genetic drivers, if there are any, that we could target for therapy? Are there any useful druggable targets in these tumors? So we think so, and we think that probably it's because of gene copy number changes that are driving some of these tumors. So this paper from 2013 points out that if you look at human tumors in general, some are dominated by high levels of recurrent mutations, point mutations, and some are dominated by high levels of recurrent copy number alterations. So these are called the M-class tumors, and these are called the C-class tumors. So these C-class tumors often have an underlying P53 mutation. There's a large number of copy number alterations per case that affect large regions of the genome. So these C-type tumors are a challenge for us to understand what really are the genetic drivers, because there's many potential candidate drivers, because the genome is completely disrupted. Many genes are undergoing copy number alterations. Some cancer genomes have very few genetic alterations and might be caused primarily by epigenetic changes. An example of this is retinoblastoma. So in this paper from St. Jude's in 2012, they found very few mutations. In fact, RB1 was the only mutated cancer gene they could see, and they concluded that epigenetic alterations are a major contributor to retinoblastoma development. So, because of these problems, we decided to try to focus on developing new ways to define the meaningful changes that have occurred in a cancer genome. Whether the tumor is dominated by genetic or epigenetic drivers, these C-class tumors and those dominated by epigenetic alterations are a challenge for defining specific drivers. So in my lab, we looked for a functional genomics approach that we could use instead. So to understand cancer development and cancer progression. And we wanted to do this by making mouse models of the human cancers we were interested in, but make them in such a way that we could identify the drivers. And then once having identified those drivers in the mouse model, ask if they're altered in human cancer. That's the basic idea. So we wanted these tumors to be similar to those that develop in human patients, but reveal the genes that were altered during the process of tumor development. And the inspiration for this was cancer induced by retroviral insertional mutagenesis. So when I was a postdoctoral fellow, I had a background in this type of research. But when I started my lab at Minnesota, I was faced with the problem that there was no retrovirus available for inducing the types of cancer that I wanted to study, such as some of these pediatric solid tumors. So we had to develop a new approach. And for that, we used Sleeping Beauty, a cut and paste DNA transposon derived from inactive TC1 Mariner family transposon elements and created using reverse evolutionary approaches here at the University of Minnesota. So this is a cut and paste element that we could use to randomly mutagenize cells in the mouse and induce cancer. So it provides us with an unbiased system for finding tumor genes. It causes random mutations in tissues of mice. We get a tumor, and we find out what genes were altered during the development of that tumor. So Sleeping Beauty is a two-part system, the transposase protein that you express in trans, and a transposon vector. And if you put these two parts of the system in the same cell, you get this cut and paste reaction. So the transposase recognizes the inverted terminal repeats, excises it from the donor locus, and reintegrates it at a TA dinucleotide that becomes duplicated in the process. So this is the cut and paste reaction. Now you could use Sleeping Beauty for a lot of things. You can put a therapeutic cargo in here and use it as a way to achieve permanent, non-viral gene transfer and expression. So for those of you interested in gene therapy, this is a kind of tool that you could use. On the flip side, you can make this thing, transposon vector, jump around within somatic cells of a mouse and induce cancer, and use the integrated transposon as your molecular tag to find the genes that can cause cancer. So two different ways you might use a system like this. But to do this somatic cell mutagenesis with Sleeping Beauty in mice, we created mice that harbored a Sleeping Beauty transposon, and it was present as a multi-copy concatemer, anywhere from 25 to 300 copies per cell, all integrated in a single place in the genome. And then we crossed that with mice that express the transposase enzyme. Now you have a mouse that has both parts of the Sleeping Beauty system that I told you about. And in this mouse, the transposon jumps around and many, many somatic insertion mutations are occurring. Now we designed the transposon so that it could turn on endogenous proto-oncogenes or turn off endogenous tumor suppressor genes. So it's one vector that can do both things. And the way it can do that is it has a strong promoter in the middle of it and a splice donor here. So this can cause the overexpression of downstream genes. And it also has a splice acceptor in both orientations and a bidirectional poly A here so that it can disrupt genes. So it can cause gain of function insertion mutations or loss of function insertion mutations. So for example, here's a hypothetical 4-exon gene, and if the transposon lands upstream of it, it can initiate a transcript which splices to downstream exons and results in overexpression, ectopic overexpression of a gene like MYC, proto-oncogene, or RAS. Those are events that we've seen before in Sleeping Beauty-induced tumors. Or it can land in the middle of a gene and disrupt it, and disrupt a tumor suppressor gene like P10 or NF1 or NF2, and those are events that we've seen in our genetic screens. Now just to remind you, we're mobilizing the transposon from a multi-copy concatemer and it jumps to elsewhere in the genome. So we're not placing it anywhere, we're letting the transposon jump around and land at random. And in a rare cell, it's going to land near an oncogene and turn it on and initiate cancer, or land in a tumor suppressor gene, knock it out and initiate cancer. So we're letting the process happen in these mice. And the long story short is the approach works. For example, these are liver tumors that we induced by mobilizing the transposon in the liver of mice. We prepared DNA from the tumors. We amplified the junction between the integrated transposon and the adjacent DNA, and we do next-generation sequencing to obtain all the unique insertion sites. And we bioinformatically distill that down to what we call the common insertion sites. So we're distilling a great deal of genetic information down to the good stuff, so it's sort of like making whiskey. And basically what we're looking for are the genes that are recurrently altered by transposon insertion in multiple independent tumors from different mice. And we use a trick to allow tissue-specific Sleeping Beauty mutagenesis, where we use a Crelox-regulated form of the transposase enzyme transgene. And by using different tissue-specific Cre drivers, we can induce different types of cancer in mice. And at this point in time, over 50 publications have come out describing Sleeping Beauty or piggyback transposon screens for cancer genes. And we've identified genes for carcinomas, sarcomas, neuroectodermal tumors, leukemia, lymphoma, and other types of cancer. Most of the screens are done in cancer-predisposed mice or cancer-promoting environment. And now we and others have started to use the screen to find genes and genetic pathways that control specific cancer phenotypes, such as the acquisition of drug resistance or the ability to metastasize. So we did a screen like this for osteosarcoma, and these are the genes that we found. So we did Sleeping Beauty induced osteosarcoma. In some cases, we used a p53 mutant background, and I'm sort of cutting to the chase and skipping all of the details. One of these screens typically takes several years to carry out, but we found well-known tumor suppressor genes that were altered by transposon insertion, like NF2, P10, NF1, and RB1. And these word clouds show the genes, and the size of the gene symbol is related to how often that gene was altered in the osteosarcomas that we induced. And in blue are the tumor suppressors, and in red are the proto-oncogenes. But what we were interested in is that some of these red oncogenes could be new targets for therapy if they were involved in osteosarcomagenesis in people. And one of those is called SEMA4D. So this is activated in mouse Sleeping Beauty-induced osteosarcomas, but interestingly, it's amplified and overexpressed in human osteosarcomas and is a therapeutic target because it's on the surface of the osteosarcoma cells and maybe is druggable using a monoclonal antibody. So here's an example of a gene that we wouldn't have been able to pick out for study. One of these amplified proto-oncogenes if we hadn't done the genetic screen. So this immunohistochemistry just shows that SEMA4D is overexpressed in a decent subset of osteosarcomas. It's one of a family of cysteine-rich sema-domain ligands that bind to receptors called plexins. And so human osteosarcomas express this. If we force SEMA4D expression on osteosarcoma cell line, it dramatically enhances their growth as xenografts. And the monoclonal antibody inhibition reduces osteosarcoma migration, 3D proliferation, and tumor formation in vivo. So this discovery led to the first clinical trial that was motivated by Sleeping Beauty screen. And this has been the work of Brandon Moriarty, Brenda Weigel here, Emily Greengard, and a company called Vasinex, which has a humanized monoclonal antibody that binds to and neutralizes SEMA4D. So that's one example. Another example that came out of the screen is colony-stimulating factor 1 receptor, which we found to be activated by the transposon in Sleeping Beauty-induced osteosarcomas, again is amplified and overexpressed in human osteosarcomas and is a therapeutic target. And to cut to the chase, the pharmacological inhibition of this receptor blocks human osteosarcoma migration, 2D and 3D proliferation, and tumor growth in vivo. So this is another example of a target that came out of the genetic screen we did in mice. So for this last bit, I want to talk about this second concept for finding new therapies, which is the idea of synthetic lethality. And we've applied this to the study of tumors that develop in the context of neurofibromatosis type 1 syndrome. So this is an autosomal dominant disease, affects 1 in 3,500 individuals. All patients with NF1 have Kaplan-Meier spots and dermal neurofibromas and benign hamartomas, Lisch nodules in the iris. They may also have skeletal abnormalities and learning and memory problems, but of interest to our work, they're prone to benign and malignant tumor development. In fact, the most common cause of mortality associated with the disease in NF1 patients is malignant peripheral nerve sheath tumor, or MPNST, a soft tissue sarcoma of the Schwann cell lineage. Now, patients who have this disease have inherited a loss of function mutation in the NF1 gene, which encodes neurofibromin, a GTPase activating protein for RAS. So this is a negative regulator of RAS signaling. So when it's lost from cells, you get too much RAS signaling, and that's what drives cancer. So these NF1 syndrome-associated peripheral nerve sheath tumors are major problems. The plexiform neurofibroma is a benign Schwann cell tumor that's congenital. The tumor Schwann cells show loss of the wild-type NF1 allele. So because the patient has already inherited one mutant copy, when the second one is lost, now there's no neurofibromin expression in those Schwann cells. And these tumors can cause major problems, and they can grow to very large sizes. Even worse, these plexiform neurofibromas can transform into the malignant peripheral nerve sheath tumors. This very aggressive, poor prognosis, soft tissue sarcoma, for which we need new therapies. Now, the process of the formation of the plexiform neurofibroma and progression to MPNST is understood in a little bit, in the sense that we know that loss of the second copy of NF1 and other factors leads to the development of the neurofibroma. And then that neurofibroma can progress to MPNST, and that is accompanied by loss of certain other tumor suppressor genes, like CDKN2A or P53. And a more recent observation is that almost all of these tumors have biallelic inactivation of SUZ12 or EED. And these two proteins are critical components of a transcription complex, regulatory complex in the nucleus called polycomb repressor complex 2. And the job of this complex is to methylate histones, in particular to methylate histone H3 at lysine 27. So without polycomb repressor complex 2, you lose histone H3 K27 trimethylation. This is normally a repressive mark. So it says that during the development of the MPNST, there's a major change in the epigenetic regulator. Okay. So neurofibromin, I mentioned, is a GTPase activating protein, or GAP, that's expressed ubiquitously, but at highest levels in neurons, Schwann cells, and oligodendrocytes. It's a very large gene. Many different mutations are found in NF1 patients. What they have in common is that they're all loss-of-function mutations. And this is a GTPase activating protein for NH, K, and RAS. And so it binds to and greatly increases RAS's GTPase activity. So RAS is a weak GTPase. When bound to GTP, it can interact with downstream effectors to signal. If neurofibromin binds to RAS GTP, it stimulates its GTPase activity. That third phosphate on GTP is cleaved off, and now you have inactive RAS GDP. So you can imagine then that if NF1 is lost from a cell, RAS GTP levels are going to accumulate, and you'll have too much signaling. But we think RAS hyperactivation alone is not sufficient for tumor development. Nevertheless, RAS-activated signaling pathways are likely to be useful for therapy. So for example, we know that RAS GTP can signal through PI3 kinase and the RAF-MEK-ERK signal transduction pathway. And in fact, the MEK inhibitor selumetinib is active for treatment of plexiform neurofibroma. So in this paper from 2016 in New England Journal of Medicine, the authors showed that treatment with a MEK inhibitor called selumetinib caused confirmed partial responses in 71% of patients. And you can see an example here where this plexiform neurofibroma shrunk dramatically in size. And this is a huge milestone in the treatment of plexiform neurofibromas. But what questions are left? Can we improve this response so that it's a complete response? Will the MEK inhibitors be useful for treating the malignant derivative of the plexiform neurofibroma? The answer so far seems to be no. And importantly, for what I want to tell you about, what other vulnerabilities do NF1-related tumors have because they've lost these tumor suppressor genes? So, to try to understand this, we've used the CRISPR-Cas system to make isogenic pairs of human Schwann cell lines that retain NF1 or have lost NF1, but otherwise are identical. And we thought we could use pairs of cells like that to identify the unique vulnerabilities that NF1 deficient cells have. That's the basic idea. So I told you earlier that the neurofibromin protein, which is shown here, has this gap-related domain in the middle. But a lot of other proteins have been shown to interact with neurofibromin. So when neurofibromin is lost from a Schwann cell, we can't easily predict what other vulnerabilities might become evident in those cells. So the synthetic lethal effects are likely to be highly context dependent. And that's why we did our work using human Schwann cells that we genetically engineered to either have NF1 or have lost NF1. So a postdoc in my lab, Kyle Williams, oversaw a project to create these NF1 deficient Schwann cells from a wild-type Schwann cell line that we had. And we carried out a drug and small molecule screen looking for drugs that would kill the NF1 deficient cells and spare the NF1 wild-type cells. So he used CRISPR-Cas to introduce the mutation into exon 10 of the immortalized human Schwann cells that we were working with. He was able to identify multiple clones that no longer express the neurofibromin protein, as you can see in this Western blot. These are control clones that retain the expression. These knockout clones have RAS GTPase levels that are higher than the wild-type clones, as you would expect based on the biochemical activity of neurofibromin. These knockout clones are partially transformed oncogenically in the sense that they have increased anchorage-independent growth, and some of them can grow as tumors when implanted into nude mice. So all of this made us feel good about using these clones in the drug screen that I'm going to tell you about now. So working with the Institute for Therapeutics Discovery and Development here at the University of Minnesota, we screened over 11,000 drugs and eventually distilled this down to 22 drugs that showed selectivity toward NF1 deficient cells. And these drugs fell into classes. Here's a list of the drugs. Such as topoisomerase 1 and 2 inhibitors, drugs that increase intracellular calcium, drugs that induce endoplasmic reticulum stress and cause the activation of the enfolded protein response, drugs that inhibit NF-kappa B, and MEK inhibitors also showed this trait, consistent with what we know about the biochemical activity of NF1. So again, we couldn't have predicted necessarily that these drugs might be selectively toxic to NF1 deficient cells. So the next question is, could they be useful therapeutically? So we've started testing selected drugs from that list I just showed you in genetically engineered mouse model of high-grade plexiform-like neurofibroma, pre-malignant tumors, and in human NF1 tumor xenografts that we put in immunodeficient mice. And I'm not going to show you all the data, but so far, every drug that we picked from that list is active in both models. So here's an example. One of the drugs that increases intracellular calcium is a cardiac glycoside digoxin, which is you probably know is used to treat heart failure. But it actually is very active in suppressing the growth of human malignant peripheral nerve sheath tumors implanted in mice. So this is tumor volume versus days. And here you can see untreated, animals treated with vehicle alone, or animals treated, tumors size over time, treated with digoxin. And the treatment actually reduces the tumor size at enrollment of both animals, and the animals live a lot longer. The drugs are, this is a longer list of the drugs that we've tried. They all work to some extent in the genetically engineered mouse model to prolong survival, and the same in the MPNST xenografts. One of the additional drugs that we found was by studying loss of the second tumor suppressor gene. So I told you earlier that most MPNSTs have lost S2Z12 a critical component of the polycomb repressor complex. And so we did a smaller screen for drugs that would selectively kill SEZ12 plus NF1 deficient cells. And these Western blots just show that we were successful in knocking out SEZ12 either on an NF1 proficient or NF1 deficient background. It completely eliminated histone H3K27 trimethylation, as you would expect. And basically what Kyle found when he screened the SEZ12 deficient cells is that they become sensitive to certain epigenetic drugs. So they are sensitive to HDAC inhibitors, aurora kinase inhibitors, DNA methyltransferase inhibitors. An example is verinostat, an FDA-approved histone deacetylase inhibitor. And this verinostat actually works very well in the MPNST xenografts to prolong survival. But these results are clearly not good enough. Eventually, the tumors keep growing. We want a treatment, a combination that will completely eliminate tumor and prevent it from coming back. So we're starting to combine the drugs. That's a simple-minded idea. So with traditional chemotherapy, that's what's done. You combine chemotherapies. And maybe one chemotherapy works in cells in S phase, and another one works in cells that are in mitosis, and you get synergy. So these drugs we're finding might be synergistic in analogous ways. So we're combining them. So for example, the MEK inhibitor selumetinib, we find is synergistic with verinostat in SEZ12 deficient MPNST cells. So we're now combining the drugs that are synergistic in vitro, we're combining them in vivo. And so a triple drug combination looks very promising. So here's tumor volume versus days after enrollment, untreated triple drug combination completely crushes the tumors. So this is the waterfall plot showing that. So what we're seeking is sustained complete tumor inhibition, and we're testing in several patient-derived xenografts next. All right, so basically what I told you today is that there are useful drug targets that you could find in copy number or epigenetically altered oncogenes. And I told you about SEMA4D and colony-stimulating factor 1 in osteosarcoma. And then this concept of synthetic lethality, loss of certain tumor suppressors, has revealed digoxin and other drugs that are selectively toxic for NF1 deficient cells, and HDAC inhibitors and DNA methyltransferase inhibitors that are selectively toxic for tumor cells that have lost polycomb repressor complex 2. So this slide shows some of our collaborators and sources of funding. In particular, Brandon Moriarty, who's now an independent faculty member, and his student Brandon Smeester have collaborated with us on the osteosarcoma work I told you about, among many others. And I wanted to mention Kyle Williams, Brian Keller, a graduate student, and a technician, Alex Larson, who I see is listed twice, because he's that special, for their work on MPNST that I told you about. And I'll stop there and take any questions that you have. Thank you.
So I just want to thank David for a tour de force, and just to say this is really the quintessential example of translational research, really using the lab, and David is a PhD partnering with many, many people to bring these amazing technological advances from the lab to the clinic. And so it's been a real pleasure to have David really bring these ideas forward and be part of our group. And you might have noticed that the infrastructure that Mark started, the Children's Oncology Group, was really the vehicle with which we took this to the national stage. So thank you, David.
David, interested in, you mentioned the FDA-approved approach of whole exome targeting. Is the work that you're doing suggesting that there could be an epigenomic approach to the same process? Study of the epigenome that could identify targets.
Yeah, I think so. So that you could imagine doing a test where you're looking for the hypermethylation and inactivation of certain tumor suppressors, and based on the synthetic lethal idea, there would be certain therapies you'd pick then, or a region that's hypomethylated and you get proto-oncogene overexpression as a consequence, and then that would suggest a certain treatment. You could use other approaches, though, just immunohistochemistry, to find whether a target is present or not. It's really figuring out which ones are the tumor cells really relying on. That's important. But yeah, so we did find that some of the proto-oncogenes I talked about are hypomethylated, and that might be their mechanism of activation in some of the tumors we've studied.
And for the NF model in particular, is there something about tumor suppressor genes that specifically affects the epigenome or not? Is there a relationship?
So a good example is with SEZ12. So if it's mutated and no longer expressed, the polycomb repressor complex can't do its job. It's no longer methylating histones, and that normally represses transcription. So you're getting inappropriate activation of gene expression all over the genome. But we don't know which genes are the ones that are most important that are affected. And so right now we are doing RNA sequencing and ChIP-seq experiments to find out what are the regions that are most affected by loss of polycomb repressor complex 2 during MPNST development.
Is NF1 somehow predisposed to SUS2
Not that we,
loss? Okay.
That's a great question. Not that we know of, but the effects of losing both they cooperate during the development of the tumor. So.
I'm out of this area. You're way beyond me. But first of all, these compounds that you're talking about, what are they? What are those compounds?
Good question. So I should have elaborated a little bit more about that. The 11,000 drugs were from various libraries of drugs and small molecules that we picked out with the help of Dr. Gunby Georg here at the university. And it included all FDA-approved drugs for any disease. And it included certain classes of drugs that are compounds that target protein classes. Like we had a bunch of kinase inhibitors. We had a bunch of drugs that inhibit G protein coupled receptors. Where there are, we chose drugs where they're likely to, drugs that are small molecules that inhibit proteins that have been shown to be useful targets in other settings. But the idea of using all FDA-approved drugs is that we might be able to repurpose drugs. We're interested in moving our findings from the lab into the clinic quickly with the help of Chris Mortel, who follows the patients who have NF1 syndrome here at the university. And we thought that if we're screening FDA-approved drugs and we find some drugs, FDA-approved drug combinations that work, it'll be easier to get into the clinic faster. So that was kind of the logic behind the choice of the 11,000 drugs.
Well, when we started with different drugs to treatment of successful different tumors that you're talking about, that we started with phase 1. And what it was, if you gave that drug or a combination of those drugs, did they have any side effects? Not about the treatment, not of the drug, of the tumor, but did it cause problems with the patient? And I have no understanding what these drugs you're talking about. But obviously, it's an area, we treated those patients with, well, they didn't work, did not work, nothing. So I think that you're right, but you now start all over with these things, but I have no understanding of what kind of these drugs are as the human body responds to it.
Yeah, so you bring up an important point. Because we've already decided that we're going to have to do some kind of combination of these drugs, toxicity will be a major concern. Some of the drugs that we're interested in based on these studies have been given together in other patients. Maybe not pediatric patients, but in adults. For example, digoxin and selumetinib have been given together in melanoma, and that's a combination that seems to be active in the MPNST xenograft. But I think this is a major concern, and to go forward to phase 1, we're going to have to we may have to do some large animal tox. But, we're taking a guess as to whether we might see unexpected toxicities or not.
That was a great talk, David.
Thanks.
You anticipated one of my questions. One of those repurposed drugs, DIG. Who knew, right? Yeah. I mean, hundreds of millions of people for centuries have been on this medication. I wondered if there was any dusty old manuscripts on linking chronic congestive heart failure with a protective effect against cancer. Any epidemiologic data at all linking long-term DIG administration or CHF with a protective effect against malignancies?
There was a study that showed that children who had been on DIG later had fewer tumors than you might expect. So yes, there is some study. I don't know, I'm struggling to remember the details on this, but I think it was Chris that pointed this out first. Do you remember, Chris?
Mark, it's James Moeller, Personal Communication.
Okay. So it is...
But yeah, it came from his study of congenital heart disease survivors, and it was his recollection and his unpublished recollection, darn it, that indeed these kids had a lower incidence of cancer later in life than the general population. I wish it was published, and I wish we could take it to justify what we're doing. Yeah. But again, as David has said, DIG has already been repurposed for melanoma, and other laboratories across the country have shown that DIG is indeed a great drug to fight cancer. So I think, we found out on our own that we're on the right track.
All right, thanks a lot.
Thanks again to David, and thank you for coming. I think he took my... Thank you.
You have been disconnected from the meeting.